Welcome to the Mitosis and Meiosis Lab video for Biology 130A Lab. For this week, we're going to ask you to watch the video and answer some questions on the worksheet. Go ahead and pause the video or review the video as needed in order to fill out the complete worksheet. Once you've completed the worksheet, be sure to bring it with you to lab. In order to participate in this week's lab, you're going to have to complete watching the video and then fill out this worksheet and bring it to lab, along with your lab handout, which you'll use to complete the lab activities. So to begin this lab video, let's go over the objectives for what we would like to cover today. First, in this lab, we'd like to learn all about the cell cycle, all of its phases and subphases. Then, we also want to understand the role that mitosis plays in somatic cells. In somatic cells, we're going to have cells that are going to help with growth and maintenance of the tissues of your body. We're also going to want to be able to describe the specific stages of mitosis, and for this we're going to use the video that we're watching today, along with some hands-on activities in lab. We also want to understand the role that meiosis plays in genetic inheritance. Meiosis is a slightly different process than mitosis, and it's going to be involved in reproduction, genetic inheritance, and in particular we want to pay attention to how recombination plays a role in sexual reproduction. We want to describe the processes of meiosis, including all of the steps in meiosis. And again, for this, we'll watch a video here today, as well as do some hands-on activities in the lab. And finally, we're going to go into the lab, and we're going to model the inheritance of certain types of traits known as Mendelian traits. We will be using pipe cleaners to represent chromosomes that carry genes, and these genes will have different versions of the genes that we'll use to watch inheritance occur. We'll apply this inheritance process to potato heads, and we'll watch potato heads engage in sexual reproduction and participate in the inheritance process. We'll watch how their traits change as they engage in sexual reproduction. Let's begin by reviewing some aspects of cell division. Organisms use the process of cell division to increase their number of cells. We would want to increase the number of cells for a couple of reasons. First, it could be for growth, as an organism is growing and developing. It can be used to maintain tissues and cells that are already in place, and cell division is also used in reproduction. The process of growth and maintenance are associated with a process known as mitosis and occurs in somatic cells. The process of reproduction is associated with a process known as meiosis, and this occurs in sex cells which form gametes that can be used in fertilization and sexual reproduction. Let's review the cell cycle. The cell cycle are the series of steps that the cell must go through in order to complete cell division. The cell cycle is initially divided into two phases. First is interphase, which is the longest phase, and it's made up of three subphases, which we'll review in a second. There's also a shorter phase known as the mitotic phase, and in the mitotic phase there are two components. There's mitosis, in which chromosomes are organized. So for example, the purple items are the chromosomes here in this cell, and they're being organized and sorted and then separated into two halves or two groups as they get prepared to be produced into two daughter cells. In order to complete the production of two daughter cells, there also has to be cytokinesis, the second part of mitotic phase, and this is what allows cell division to be completed as the cytoplasms of the cells are completely separated. Now, if we look closer within interphase, we'll see that there are a series of subphases. First is the G1 phase, which occurs right after cell division. The G1 phase is a growth phase that allows the cell to grow into its new functional form. Now, the G1 phase may stop at some point and stop the dividing state. At this point, we reach what's called the G0 phase. The G0 phase occurs when the cell is no longer getting signals to divide, and it can simply undergo its normal cell function in a non-dividing state. If the cell does get a signal to continue division, because more cells are needed, then it will continue on into the S phase. In the S phase is when DNA replication occurs, and each chromosome in replication is copied during this phase in order to form identical sister chromatids, which we'll discuss in a second. If the cell continues past S phase, it goes into G2 phase, which is a second growth phase, and this growth phase is preparing the cell for cell division, in which case this one cell will have to become two cells and will need enough cellular machinery and material to produce two daughter cells. To better understand mitosis and meiosis, we need to know a little bit about chromosomes. Chromosomes can be viewed in a couple of ways. First, they can be viewed as a linked array of genes that code for the production of certain types of cellular products. But they can also be viewed as microscopic structures that are made up of DNA, a nucleic acid, and protein. Chromosomes are typically described by a combination of their size and their centromere location. The centromere is a little pinched part of the chromosome structure that will be used as an attachment site during mitosis and meiosis with microtubules or spindle fibers. In the laboratory, if you stain the chromosomes appropriately, you can actually see banding patterns that are unique to each type of chromosome. Now, in most somatic cells, we have a diploid condition in most organisms. A diploid condition means that there are two chromosomes in each cell of each type of chromosome. So, for example, in this picture, we see a karyotype, which shows two types of chromosome 1, two versions of chromosome 2, and so on and so forth. Those chromosomes that match each other are considered to be homologous chromosomes. So in a diploid organism, each chromosome will have a partner, a homologous chromosome that carries the same sets of genes. This is why the banding patterns are similar. Each of these two homologous chromosomes then will come from a different parent. So in the case of chromosome 1, one of the chromosomes may have come from the father, whereas the other chromosome 1 may have come from the mother. Humans have 23 different types of chromosomes, and they're diploid, so they have two copies of each of those 23 types of chromosomes. Cells can also have a haploid condition. Haploid cell is a cell that has only one copy of each chromosome. The haploid condition can be found in gametes. Examples of gametes include sperm and egg. Now, sperm and egg, as gametes, can get together in a fertilization event, and when they get together in a fertilization event, they can combine their sets of chromosomes to form a diploid zygote, which can then undergo cell division to become an offspring. We also need to understand a little bit about what's on the chromosomes. These include genes and alleles. So genes are sections of DNA on a chromosome that have a code for the production of a specific type of molecule, whereas an allele is a version of a gene. So genes can have different versions. So for example, in diploid organisms, you can have two chromosomes, and on each of those chromosomes you can have a gene that codes for a certain product. And in this case, the alleles, or the versions of those genes, are identical. When the alleles are the same, we call this a homozygous condition. In another organism, we could have two chromosomes that carry that same gene, but in this case, they carry two different versions of that gene, or two different alleles. We call this condition heterozygous. These genes then are going to combine to form genotypes. The genotype in a diploid organism is when you have a combination of the two alleles. As we just saw in these two different organisms, they each had two different types of genotypes. One had a homozygous genotype and one had a heterozygous genotype. It's this genotype that will determine the phenotype. The phenotype is a version of a trait or some collection of traits. The phenotype is what the organism is like. It's the observable, physical, or biochemical characteristics of that organism. So for example, this genotype, which is the homozygous condition, may code for some gene product that causes ears to be a bit stiffer and to stand up. Whereas the heterozygous form may have an allele or a version of the gene that causes ears to not be able to stand up and become in the droopy condition. So the genotype would be homozygous or heterozygous in this case, but the phenotype would be how do the ears look. Now, let's watch a short animation that will allow us to visualize the process of cell division and the specific steps of mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Now be sure that you have answered any questions on the worksheet related to the mitosis video. Now let's watch another animation about meiosis and understand the specific steps involved in meiosis.
Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with two sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Now be sure that you've answered any questions on the worksheet related to the meiosis video. Let's talk a little bit about what we're going to be doing in lab this week. So the first lab activity, we're going to work in small groups, and it's going to be an activity about cell division. In this case, we're going to model the stages of mitosis and cytokinesis using some items we can find around the house so that we can really get a good idea and review what we just learned in the video about the steps of mitosis. Again, work in about small groups of about 4 students and go collect a mitosis kit from the instructor. In that kit, you'll find various bits of string and pipe cleaners and some spools that will represent different cellular components that are involved in the cell division process in somatic cells. The chromosomes will be represented by pipe cleaners. Pink twine will serve as the cell membrane. White twine will serve as the nucleus membrane, or the nuclear membrane, and there'll be some spools in there that will serve as centrosomes that we just met in the video. There'll be a bead which will serve as a centromere for our chromosomes, and then there'll be some wire on the spools which will serve as microtubules or spindle fibers, which will be involved in organizing the chromosomes. Follow the directions in the laboratory handout once you get into lab and complete this activity. Work through each of the stages of interphase, mitosis, and cytokinesis together. For the second laboratory activity, we'll be comparing genotypes and phenotypes. For this laboratory activity, we'll be working in small groups of 4 again, but in this case, the groups will all work together in a step-by-step manner guided by the laboratory instructor. You'll need to, when you get into lab, you'll need to retrieve a chromosome packet from the instructor, and in that packet you'll find pipe cleaners with letters on them. The pipe cleaners are chromosomes, and the letters represent genes. You'll notice that you can have different versions of the letters, and that means that the genes can have different versions of the genes known as alleles. You're also going to need to retrieve a potato head from the instructor. Potato heads have a variety of different traits, and what we're going to notice is that each combination of alleles or each genotype that you have in your packet will code for a specific phenotype or look for the potato head. The genotypes on the chromosomes will determine which phenotype your potato head will have. You're going to follow the instructor's guidance as well as the instructions in the laboratory handout throughout this entire activity. And part of what you'll be doing is mating potato head from your group with those from the other groups. You'll want to record the results of each mating after examining the offspring and seeing how the phenotypes end up when comparing their genotypes. Examine the results of meiosis and of recombination, which is the ways that the different alleles get recombined into different combinations to produce very different phenotypes. Once you've completed this video, be sure that you bring your completed worksheet to lab. This worksheet is going to be your ticket to getting into lab and being able to participate in the lab. Again, in order to participate in this week's lab, you'll have to have your completed worksheet along with your lab handout with the laboratory instructions. We look forward to seeing you in the lab. See you later this week.